Assalamu alaikum viewers. Hope for best of your health and iman. Today's topic is transduction. Transduction is the third process of reproduction, sexual reproduction in bacterial cells, which is our today's topic. We're going to discuss this topic, transduction. Transduction is that process of reproduction in which bacterial cell transfers genetic material from one donor cell to the recipient cell with the help of a third party, which is usually a bacteriophage or a phage. So this is a process of transduction in which a third party is involved for the reproduction or for the transfer of genetic material from donor cell to the recipient cell. It has been discovered by the Zinder and Ledberg in 1952. So this is about the history. Okay, what happened, how they discovered the transduction, and some circumstances behind all the discoveries of science and everything. There is some some story, a coincidence of anything which they discovered about anything. So here was also one story that they were working with a salmonella, typhimurium, which is a bacterium which causes typhoid in mouse. So they were working with this cell and studying about the bacterial conjugation. But what happened during this process, they came to know about transduction, which is the third product of reproduction in bacterial cell. So they were working with salmonella, and they were studying about the this bacterial conjugation. So they were about studying the bacterial conjugation, checking out or investigating the bacterial conjugation. And what happened, they had the mutants. They produced the mutants that some of the nutrients, and they were that kind of the nutrients. They mutated the cell like this, that they were not unable to produce these nutrients. First of all, they were unable to produce these nutrients. Some of them nutrients which they take off their property to produce these nutrients. So what they did, they tried to produce the that type of the mutants, bacterial cell mutants of bacterial cell, which were unable to produce some particular type of the nutrients. But when they grew them in culture and they see them, and they found out that they were able, they found the recombinants. They found some recombinants, you see. So they found some recombinants, and they see that these recombinants, these cells were able to produce all of these nutrients. So what changed these bacterial cells? What enabled them to produce all these types of nutrients? But they did not they produces these bacterial cells in this way that they could not produce. So results was again unexpected results. All three processes we have discussed about conjugation, about transduction, about transformation. So all results, they get unexpected results. So what happened, they found recombinants, somewhere mutants of recombinants variety, which were able to produce all of these nutrients. Now they found out that there is something happened in this case, and something have occurred at this moment that make them, these bacterial cells, able to produce all of these nutrients. And they found out that there is some phage, or the bacteriophage, a phage virus, a bacteriophage is responsible for this process, which enable them to produce all of these nutrients. And how they found out that, and what is the mechanism of this process of transduction? So let's have a look on of these. It gets attached itself to the bacterial cell, and when it gets attached itself to the bacterial cell, the all process bending or release down of its DNA. So it gets injected its all of DNA into the bacterial cell. So when DNA is injected into the bacterial cell, then again there will be two possibilities. One is that in which the bacterial cell synthesize its own protein, its own components, its own required substances, all of it, which is known for its survival. And in this way phages are produced, and then phages are assembled. All the new phages are assembled, and bacterial cell at the end burst, cell wall burst, and bacterial cell release all of its phages. So new assembled phages are released out of the cell, and in this way they're gonna attack all other bacterial cell. And in this way it is a third party, a vector formed between the transfer of genetic material from one donor cell to the recipient cell. So in this way they do all this. And the second process that is the lysogenic cycle. If you want to know all of about lysogenic cycle and lytic cycle, so here is the link I have posted in, so you will be able to know about this link all the detailed video about the lysogenic cycle as well as lytic cycle. So you can easily found out all of these processes how it takes place. So this is the methods of lysogenic cycle and lytic cycle. And what happened in the lysogenic cycle? In lysogenic cycle, the bacterial cell get integrated its DNA into the bacterial DNA. So phage DNA integrated into the bacterial DNA, and it gets alongside replicates with the bacterial DNA, its bacterial chromosome converted into the all of its process, and replicates and produces it's when the bacterial cell replicate, it produces progeny as well, progeny of phages, phage DNA. It is produced alongside the all of the bacterial cells, and this bacterium which get integrated, it is known as prophage. And the virus which produces all of this process is known as temperate virus. And the bacterial cell in which this process takes place is known as lysogenic bacterium. So these are the terms which should be in our minds, in your mind, that how the terms are related with this process. So the bacterial virus then produces its copies, and bacterial cell, and there will be two types of transduction. One is generalized transduction and one is specific transduction, specialized transduction. I'm gonna discuss all of these generalized transduction and specialized transduction in next episodes. So stay tuned for all of these important and helpful videos. And they got the change in the chromosomes, and in this way bacterial cell get changed, and its DNA, its chromosome is changed and variety is changed. And this is in this way phage act as a vector between all of these. Okay, how they found out all this transduction process? They perform a tube experiment, a transduction tube experiment. What did they do? They form a U-shaped tube, and in two arms of a U-shaped tube, in a U-shaped tube like this, U-shaped tube, in both the arms of U-shaped tubes they put the bacteria. So when they have the you see here is like this, a U-shaped tube, and like this, here is bacterial cell, here is two arms they filled up with the bacterial cells, and here they put one like a filter which is in which they could not be able to filtrate the bacteria. So bacteria could not pass from these filter material which they put here, and in this way there will be no transfer of the genetic material. So in this way transformation ruled out, because in transformation there is something transfer of the genetic material. No conjugation, because there will be no contact. So these two products, now it means that there is something happened which is third form is a new type which is happened, and is that form that one from the bacterial cell lytic cycle or lysogenic cycle act in some of the bacterial cells, and then they transfer their genetic material from one type to the other type of the cells. And in this way from this filter type, this filter plate, the phages are able to pass out from this filter material, this filter tray, and in this way they pass out to other bacterial cell, and they found that type of the bacterial cells which are which would be able to produce the nutrients or mutants and the other characters which can form easily. So in this way they found out the transduction. So I tried my best to explain all the topic in in my best way, that I hope it will be helpful for you. And thank you so much for coming on my channel. Don't forget to like, share, subscribe and comment, and make sure to follow me on Instagram. And bye bye, Allah Hafiz.